The cell cycle. The cell cycle is the ordered series of events required for the faithful duplication of one eukaryotic cell into two genetically identical daughter cells. In a cell cycle, precise replication of deoxyribonucleic acid or DNA replicates each chromosome. This is an animal cell. An animal cell is composed of three landmarks: the cell or plasma membrane, a cytoplasm, and the nucleus. Cells in our body can either undergo mitosis or meiosis. Somatic cells like our nerve cells, skin cells, and blood cells undergo mitosis, whereas gamete cells like egg and sperm cells undergo meiosis. Today we will learn about how this occurrence in our body happens. Let us discuss first the mitosis. It is a process where single cell divides into two identical daughter cells. It is also responsible for the growth and repair in our body. Before the mitosis phase happens, cell will first undergo interphase, where the cell prepare for division. Interphase proceeds in three stages: the gap 1, synthesis, and gap 2 phase. The gap 1 phase is the period between mitosis and S phase, when each chromosome has one chromatid. Cells spend most of their time in gap 1 phase. It is the time when the cell grows and performs its normal function. Synthesis is the time when the DNA is replicated, when the chromosome goes from having one chromatid to having two chromatids held together at the centromere. Gap 2 is the period between S phase and mitosis. The chromosome has two chromatids and the cell is getting ready to divide. M phase includes division of the cell nucleus or mitosis and division of the cell cytoplasm or cytokinesis. Mitosis is a nuclear division that results in two new nuclei, each having the same number of chromosomes as the original nucleus. The parent cell is the cell that divides, and the resulting cells are called daughter cells. Mitosis is subdivided into four recognizable stages: prophase, metaphase, anaphase, and telophase. In prophase, chromatin in the nucleus begins to condense and becomes visible as chromosomes composed of two sister chromatids that are pinched together at the side of the centromere. Special proteins attach to the centromere to form kinetochore. Finally, the nuclear membrane disintegrates and the nucleolus disappears. Centrioles begin moving to opposite ends of the cell and the fibers extend from the centromeres. Some fibers cross the cell to form the mitotic spindle. Next to prophase is metaphase. By the end of prophase, the spindle fibers align the chromosomes along the middle of the cell nucleus by their centromere. This line is referred to as the metaphase plate. During this stage, the sister chromatids appear to repel each other, forming the familiar X shape of a chromosome. This organization helps to ensure that in next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. After metaphase, next is anaphase. At the onset of anaphase, enzymes break down the proteins in the centromeres, allowing the two chromatids to separate. Once the chromatid separates, they are considered to be individual chromosomes. The paired chromosomes separate at the kinetochores to move to opposite side of the cell. Motion results from the combination of kinetochore movement along the spindle microtubules and through the physical interaction of polar microtubules. The last stage of mitosis is telophase. In telophase, this is when the chromatids reach the opposite poles of cell. A new nucleus begins to reform around the chromosomes, forming two daughter nuclei. Inside the nuclei, the chromosomes begin to uncoil and the nucleoli reappear. Outside in the cytoplasm, the mitotic spindle disappears, leaving the centrosomes just outside the nuclear membranes. The chromosomes disperse and are no longer visible under the light microscope. The spindle fibers disperse. And cytokinesis, or the partitioning of the cell, may also begin during this stage. Once the nuclear membrane has reformed around the nucleus, mitosis is complete. Cytokinesis is the final step of cell cycle. Once the nuclear contents have been duplicated, the cytoplasmic contents such as organelles, proteins, membranes, and cytoskeleton must be divided. Let's proceed to meiosis. Meiosis occurs in eukaryotic organisms that produce sexually. This includes plants and animals. Meiosis is a two-part cell division process that produces sac cells with one-half the number of chromosomes as the parent cell. There are two stages or phases of meiosis: meiosis 1 and meiosis 2. At the end of the meiotic process, four daughter cells are produced. Before a dividing cell enters meiosis, it undergoes a period of growth called interphase. Just like in mitosis, interphase in meiosis also take place, wherein the gap1 phase is the longest phase of interphase and the cell increases in mass in preparation for cell division. In synthesis phase, DNA is synthesized. In gap2 phase, the cell synthesizes proteins and continues to increase in size. Keep in mind that before meiosis begins at all, the DNA undergoes replication, just like it did before mitosis started. After interphase 1 in meiosis 1, let us proceed in prophase 1. Just like in mitosis, during prophase, DNA condensation occurs. The nuclear envelope and nucleoli disappear, and the spindle starts to form. The big difference is what is going on with the chromosomes themselves. Crossing over or crossover requires that the chromosomes break and reconnect to the other chromosome. After prophase 1, metaphase 1 takes place. Tetrads line up at the equator. The spindle has completely formed. During prophase 1 and metaphase 1, the genetic recombination is occurring. After metaphase 1, anaphase 1 occurs. Tetrads pull apart and chromosomes with two chromatids move toward the poles. And finally, the cell undergo telophase 1. Chromosomes with two chromatids decondense and a nuclear envelope reforms around them. Each nucleus is now haploid. At the end of meiosis 1, each chromosome still had two chromatids, the amount of DNA that a cell should have. So, the entire reason to go meiosis 2 is to reduce the amount of DNA back to normal. Basically, to split so that each daughter cell has only one chromatid per chromosome. In meiosis 2, the interface is similar to mitosis. The only difference is that the two chromatids per chromosome are not necessarily identical due to genetic recombination occurring in meiosis 1. In prophase 2, chromosomes with two chromatids become visible as they condense, and the nuclear envelope and nucleoli disappear, and the spindle is forming. In metaphase 2, chromosomes with two chromatids line up at the equator. The spindle is fully formed. In anaphase 2, chromosomes split, so that a chromosome with only one chromatid heads toward each pole. And finally, the cell proceeds to its final stage, the telophase 2. Chromosomes with only one chromatid condense and get surrounded by new nuclear envelopes. The four daughter cells are now all haploid and have the right amount of DNA. They are ready to develop into sperm or eggs now.